If everyone could take their seats, we're going to get started. I'm Sharon Terry. I'm chair of the Planning Committee, and I'd like to welcome everyone on behalf of the Planning Committee. This meeting is hosted by the Institute of Medicine's Forum on Drug Discovery, Development, and Translation, the Forum on Neuroscience and Nervous System Disorders, the National Cancer Policy Forum, and the Roundtable on Translating Genomic Basic Research for Health. The membership of the forum and roundtables, as I think most of you know, offer an ongoing neutral venue where stakeholders in government, academia, industry, foundations, consumers, patient groups, the public, etc., can meet to discuss and confront issues of personal interest. These forums educate the public, the press, and the policy community. Through the conduct of these workshops and other activities, these groups bring together thought leaders, and many of the thought leaders in the area of data sharing will be with us today. In accordance with the National Academy's procedures, an independent planning committee organized this workshop. They did a lot of work, I can tell you, to bring together a really stellar group and to put together, I think, a very cohesive event for all of us. The planning committee worked over the last 4 months through conference calls and emails to develop these key topics and areas of clinical research data. I'm sure none of the planning committee thought they would be working this much for a workshop, but it really is well worth it. In terms of the format for this 1.5-day meeting, we'll begin with a discussion of the benefits and risks of sharing clinical research data and suggest why and when increased data transparency or sharing can improve scientific knowledge and move forward public health. Session 2 will consider various data models and distill the best practices and lessons learned from each project so that we can build better data sharing collaborations in the future. To close this day, the first day, Session 3 will consider the opportunities and limitations of data sharing, standardization, and governance models. Friday morning, tomorrow, we'll begin with Session 4 and a discussion of the incentives for and against enhanced data transparency and sharing across all sectors. And our closing Session 5 will include reports from the session chairs regarding key topics highlighted in each of their sessions and identifications of issues that warrant deeper and more focused consideration. We'll finish the meeting with a panel discussion to help consider resolutions of the thornier issues that come up over the 2 days and highlight strategies that represent low-hanging fruit which could be acted upon quickly. As you can see from the agenda, which I hope all of you have, and if you don't, there's some out at the table, the workshop includes presentations by members of the federal government, the private sector, academia, patient community to consider the benefits of and barriers to clinical research data sharing. We look forward to robust participation from the speaker, the panelists, and the audience in identifying potential strategies for enhancing data and transparency. This is not going to be a show and tell. This will be very participatory, and we expect that all of you have something to say about these issues. The word transparency is really important for this workshop. We're really asking that each of us model that. If we expect to move forward public health and advance research, particularly through transparency, then each of us needs to be transparent and needs to be able to name the elephants in the room and work on the thorny issues together. This is a standalone workshop, and it's not part of an ongoing study by the IOM. A summary of the presentations and discussions during the workshop will be prepared by our rapporteur and will be reflecting what transpires here. So it's important to put on the record what we need on the record, and so we need to hear from all stakeholders. It will be prepared in accordance with the institutional policies and procedures and will be published by the National Academy's Press and made broadly available to the public. All views represented in the report are those of the workshop participants, and the report will not contain any findings or recommendations by the Planning Committee. We are leaving this open and very participatory. In terms of just some housekeeping, the workshop is being webcast live. More than 200 people have registered for the webcast. We're in this room. We're also in an overflow room. We'll be taking questions from the webcast audience via email, and we encourage those of you who are participating remotely to really participate. Send your questions in to us or comments, and they will be given to the chairs and to me so that we can make sure that they're part of the conversation. In terms of breaks and meals, today we have a morning break from 10:30 to 10:45, lunch from 1 to 1:30. You can see these are very short. We are really packing it in. An afternoon break from 3 to 3:45, and then on Friday we'll have a break at 10:30 and we'll adjourn at 12:45. Audience members, when you ask questions or provide comments, please speak into the microphone, which is in the center, middle center of the room there. panelists participating in roundtable discussions, you're going to need to turn off your microphone when you're not speaking because we can't have several of these on at once. Slides presented today will be posted on the IOM website within about 1 week, and the webcast will be available in about 2 weeks. And speaker biographies are available in the handouts provided in the registration table, which leads me to the beginning of this workshop, and that is to say that throughout the day and a half, we will be giving you very limited introduction to the speakers because we do have their biographies for you to read, and we do want to get to the meat of the matter. So we're going to begin with our first session, which is Benefits of Clinical Research Data. We have 2 chairs for this session. Dr. Deborah Zarin, who is overseeing the development and operation of an international registry of clinical trials, which I'm sure most of you have heard of, clinicaltrials.gov. She's also the Assistant Director for Clinical Medicine at Lister Hill. We also have Dr. William Potter, and Dr. Potter has been for a very long time a champion in psychopharmacological sciences and development of novel treatment for CNS disorders. He worked at NIH for 25 years or so. He also worked at Lilly Research Labs in translational neuroscience. He's championing disciplined hypothesis testing targets in humans to best move CNS drug development forward. And they will lead us in this first and basically setting the agenda for us workshop. Thank you.
Thank you, Sharon, for your wonderful leadership. I feel like I'm running a relay race, so we haven't practiced the handoff, but we'll see what we can do. Welcome, everybody. I just want to go through 2 basic things. Actually, can I advance the slides? Or can someone else advance? This is about sharing clinical research data, and in all of the planning discussions, I want to give a plea for precision in language. People talk about raw data all the time. But it turns out that raw data, probably people mostly mean something about participant level data. Some people mean the uncoded data. Some people mean essentially the analyzable data set. So there's a whole spectrum. And there's a distinction between the participant level data and summary data. So I will talk more about this list, some of which I got from Steve Goodman, who's also here, and Peter Doshi, who will also talk about this. But I'll just ask people when they talk about sharing data to actually be precise and clear about what they're talking about. And then the next thing, to make another point, which will become clear in the agenda and probably was clear if you read the introductory materials, is that when we talk about sharing research data, we're trying to make the point that although summary data are generally available through for those of us who don't work at the FDA and don't routinely get the participant level data, we all rely on summary data. They're generally available through journals, through clinicaltrials.gov and other sources. And what we're talking about in this workshop is really what are the benefits and challenges associated with sharing participant-level data. And what we realized was there are 2 types of foci, and we'll try to balance those in this workshop. One is participant-level data for the purpose of understanding the results of an individual clinical trial. What more can you understand from participant-level data as opposed to just the summary data? And then also the second purpose, which is really about pooling data across studies to gain additional information and to learn more than you could have from one trial. And in fact, our 2 speakers now will sort of will discuss those 2 types of issues. So I'm just going to briefly introduce Dr. Loader, who is a senior research editor at BMJ and edited a recent theme issue on unpublished clinical trial data. She's also on the faculty at Harvard Medical School. So here we go.
Thanks very much, Deborah, for that very nice introduction. It's a pleasure to be here this morning on behalf of the BMJ to discuss the fundamentals and benefits of sharing participant-level clinical trial data. I will aim to cover exactly what it is we're thinking about sharing. Deborah alluded to some of the problems in the definitions and the fact that different people mean different things when they speak of sharing particular parts of the data set. And I'll also aim to make at least a case for why it is we should share and what some of the benefits of sharing might be. Now, I'm here today in my capacity as a research editor for the BMJ, but I'm also here in my capacity as a doctor who treats patients and who therefore relies on clinical trial data to make everyday clinical and treatment decisions. And I'm here as someone who in the past has participated as a clinical investigator in many multi-center randomized clinical trials. So I've seen how the data are collected and some of the ways in which it can go wrong. And finally, I'm here in the same capacity that many of you are, and that is as a future patient and as the relative and friend of future patients who will rely on clinical trial data. And I think it's that last perspective that is very helpful to bear in mind as we go through discussing the possibilities and problems associated with making these data more available. So, what is it that we mean when we bandy about these phrases, such as individual participant data or individual patient data, raw data, and so forth? There is a lot of ambiguity sometimes, and people are not always clear on what exactly we are talking about. And because it's important that we have a shared understanding of the way in which data are generated in the course of a clinical trial and exactly what we might be referring to when we use some of these phrases, let's go through the process of data collection, data coding, and data aggregation that occurs in a typical clinical trial. And you can think of it as a multi-center trial where there are multiple individual sites possibly spread around the country or the globe, or a trial that takes place in one center. The problems are often quite similar. So here you see some case report forms. These happen to be handwritten case report forms. It's increasingly common now to use computerized forms and to enter data electronically. And these are paper or electronic questionnaires on which or in which the data from each trial participant in each clinical site is collected. And it's all collected on these case report forms and documented on these forms. Now, as you can see, these forms are sometimes far from perfect. Here you see scribbling in the margins and a lot of entries that are not immediately understandable. So these case report forms have to be processed and sent to study administrators or whoever is coordinating and organizing the study, but they are rarely perfect. So study monitors are hired to audit the data. They go to the individual site, or sometimes they audit the data electronically from afar, but in either case, their duty is to make sure that these case report forms contain the correct data. And when they have questions or data are missing or do not make sense, that generates a query back to the investigator or the study staff that has to be resolved before anything else happens. So these questions about entries have to be explained. For example, a weight might have been entered as 750 kilograms, which is implausible, and the study monitor might query that and say, did you mean 75 kilograms? Information might be missing. A male might be listed as being on birth control, and so forth and so on. So each query has to be explained and resolved before the data are entered into the coordinating center database. And at several points along the way, some types of data are coded or categorized, and we'll talk particularly about how that happens with adverse event data. At the coordinating center, there's a process of data entry, and we hope a process of double checking to make sure that data entry is correct. And sometimes in the process of data entry, additional queries about the data are generated. Another data clarification form has to be generated. There's another process and another round of querying back to the original investigator and the study staff. So this is an example of the process through which the uncoded data are transformed into coded data in at least some cases and then aggregated and entered into a database. And many people who speak about individual participant data are speaking about having access to the individual de-identified records that reside in that computerized database, but not always. And I think it's worth considering for a moment some of the challenges involved in coding adverse events, where some of the prior documents might be of great interest. So as you can imagine, misclassification of adverse events in clinical trials can have very, very serious consequences. And so systems have evolved to try to minimize the scope for interpretation and incorrect classification of adverse events. And adverse events are usually categorized using some sort of predefined hierarchy or system. And here you see, for example, how symptoms consistent with the common cold might be identified and categorized. Now, patients don't necessarily come in saying, I have a respiratory disorder. They come in saying they have a runny nose, their eyes are itchy, and they're sneezing, and someone has to make the decision that that represents a cold. So there's a lot of scope for interpretation along the way, particularly with data that hasn't been anticipated, as is the case with many adverse events or side effects. And one study reported that when two people were coding adverse events, about 12% of the time they did not agree in important ways. And in situations where doctors have gone back and looked at the case report forms of the patient descriptions of adverse events, about 8% of the time they feel that they haven't been coded correctly. And there certainly are more egregious cases, for example, in clinical trials involving antidepressants, where adverse events such as suicidal behavior or gestures were coded as things like emotional lability or coded as an admission to the hospital. So I think you can see some of the reason that coding generates a certain amount of concern and why some people want access to some of the earlier data. So what is it we're thinking of sharing? Well, here's a typical call for data is all data from randomized clinical trials, including raw anonymized data. And Peter Gutschke, in his editorial on this matter after the Cochrane collaboration issued its statement in favor of sharing data from clinical trials, is very specific. He says raw data about adverse events should mean the original descriptions exactly as reported narratively by patients or researchers. And he says on the case report forms before any coding or adjudication has taken place for categorization purposes. So the Cochrane collaboration has issued a typical statement advocating sharing data, and you see it reproduced here. They call for all data from all trials. They specify raw, anonymized individual participant data, and they ask for these, along with the trial protocol, to be publicly available free of charge and in easily accessible electronic formats. And as Dr. Zarin mentioned, the purpose in most people wanting access to these sorts of data is to enhance the understanding of what happened in an individual clinical trial, but beyond that, to enhance the understanding of what happened to individual participants within that trial. So here's an example of individual participant data that comes from a paper published in the BMJ that looked at issues surrounding individual participant data meta-analyses. And this happens to be individual participant data from 10 trials, but it gives you the flavor of the sort of individual participant data you might be able to glean from records for an individual trial. So if you look at the top there, in the first study, the first patient, patient 1 there, is a 46-year-old man. He was in the treatment group, and his pre-treatment systolic blood pressure was 137, and his post-treatment systolic blood pressure was 111. You can see the same data for the second participant in that trial, and you can see data for individual participants from all the different clinical trial sites if you're looking at a data set that has aggregated information for individual participants among clinical trials. Now, what's the advantage to having this detailed data? Because, of course, we could simply tote up all the blood pressures pre- and post-treatment for the two groups and present the average or the median. But, of course, information is lost. And in particular, collecting data and having data available to analyze at the level of the individual participant enables you to apply different staging or grading procedures, for example. And it allows straightforward categorization of subgroups so that you can examine whether or not there might be any subgroup benefits or harms. Here's a comparison of the power of meta-analyses done with individual patient data compared with meta-analyses on the same topic done with aggregate data, showing that as a rule, meta-analyses done with individual participant data are much more likely to be able to detect treatment effects that differ across groups, across different ages, for example, different sexes, or other subgroups. And I think all of this reminds us of that famous phrase, the title of an essay by Stephen Jay Gould, "The median is not the message." And certainly in clinical trials, very frequently one is interested in identifying subgroup effects. Now, historically, the currency of clinical trial publications and the currency of meta-analyses has been summary level data, but that is changing. And you see here a graphic depiction of the increase, the substantial increase over time in the number of individual participant data analyses. So this has become a subject of great interest to many people who realize the advantages. I want to spend a little bit of our remaining time going over some of the advantages that might accrue from sharing individual participant data. And as you'll recognize, these generally are likely to accrue sharing any sort of trial data. So one of the things that one hears about before we talk about the advantages is who exactly are we talking about sharing the data with? And it's useful to think about the continuum of sharing that is possible. So historically, there have been situations in which the only place where all of the data reside and can be examined are with the sponsor. Sometimes they have even not made available to individual researchers in a multi-center trial the data that they have generated from their own center or the data that has been generated by the trial overall. Some trial agreements specify that investigators may not necessarily have access to that, or if they do, their publications and research proposals generated from it have to be approved by the sponsor. It's also possible, of course, to decide that you'll share data with the clinical trial investigators or with other approved investigators, meaning that you might reserve the right to vet their data and decide if they're going to be doing the research and the analysis in a way that you think is robust. Or you might make it available to any researchers as long as they can show that they have a genuine research interest in this question and a track record of research capability. Or you might go further, and this is what I think many people will advocate and the goal to which we should aspire and share with everyone. And why might we want to make clinical trial data more available? Well, there are many, many advocates who have gone on record in favor of this, and here are just a few of them. And here are the phrases that resonate through their statements, and you'll recognize words like trust and respect and quality and efficiency. Individuals as well as organizations have gone on record in favor of this. And I think the reasons can be grouped into two categories, two main themes. The first is the moral and ethical arguments in favor of sharing clinical trial data, and the second theme, I think, is the practical and scientific arguments. And of course, many of the arguments transcend category and have elements of both of these arguments within them. They are, of course, very similar to the arguments that have been mounted before for other forms of transparency in the clinical trial research enterprise, namely registration of clinical trials and so forth. So I think in some ways this is the natural evolution of information wanting to be free. Another reason that's often mentioned is obligations to research participants. They participate in clinical research, at least in part, on the understanding that their data may not benefit them, but will perhaps benefit others. And how can it really do that to its full extent if it is under lock and key and can only be viewed by certain people? Certainly, Dr. Drazen in his recent editorial said we owe these people respect, not only the people who have been in clinical trials, but also, I would submit to you, the people who are going to be in clinical trials in the future. And many of you are no doubt familiar with the early phase trial that went very badly wrong in England a number of years ago. And there have been a number of commentators who have suggested that perhaps the adverse events that befell the 6 participants in this trial could have been prevented if earlier work on this compound had been published and available to people. It's also the case that many people claim that medical research is a public good. Now, President Obama landed himself in hot water a while ago when he said to business people about their businesses, "You didn't build that." In other words, that it wasn't necessarily, when you form a business, a completely private act. And I think that argument actually applies very well to medical research. Medical research is a public good. It's aimed primarily at producing a public good, which is the knowledge that's needed to attack important societal health problems. And the money argument also resonates. The fact is that taxpayers, the public, provides a huge amount of money to support basic research, a huge amount of NIH money. They support directly and indirectly the public universities where research is carried out. So I think there is an argument that the public, which then ends up paying for many of the products of that research, should be handed perhaps a better deal. We had an issue earlier this year in the BMJ, a theme issue that I handled, about what are the effects of missing trial data and what might be the effects of making it more publicly available. And one myth that I think was dispelled by a paper that appeared in that issue of the journal is very important. And that is that I think if we polled this audience or asked the man on the street, if hidden clinical trial data came to light, what do you think its effect would be on our estimates of the harms and benefits of existing therapies? I think the average person would say it would be likely to change our estimate so that we think those things are less effective. Now, in fact, that's not true. We don't really know how things would be affected if those trial data were released. We don't know the direction in which the results would move. And this paper showed that when unpublished FDA trial data were added to meta-analyses of existing therapies, only 7% of the time did the estimate not change, but when it changed, about half the time it changed to make the compound or treatment look more effective and half the time less effective. So we don't even know what we don't know until we have the trial data. Another argument is that availability of trial data will accelerate research by making available information to more scientists, and that research will be more productive and things will happen a lot faster. And certainly these social network diagrams show the extent of collaboration and cooperation among scientists already, but just imagine how much deeper and richer the connections might be and how many more of them there might be if they had clinical trial research data available to work with. Now, it's also the case, I think, that we can imagine certain scientific collaborations occurring, but there are probably stakeholders out there who would be able to contribute importantly to scientific research and make use of these data in ways that we cannot currently imagine. And I'm speaking about citizen science. Now, the former music director and conductor of the Boston Symphony Orchestra, Serge Koussevitzky, was known to utter many, sometimes I think unintentionally poetic statements because he was not a native English speaker. And one of the things he said, I think, applies here. He said, "The next Beethoven will from Colorado come." And in saying that, he was recognizing, I think, that genius and inspiration and ingenuity do not reside in the usual places, but may actually be found in places that we do not expect. And here you see, I think, how that could be transcribed to apply to a situation where a high school student came up with a very interesting scientific project and was able to say that it happened because he had access to freely available online scientific information. So how many collaborators are there out there that we can't even imagine at this point who might make use of the data? And the final reason that I want to advance for sharing clinical trial data, individual participant data, as well as general data, is that it would help restore trust in the clinical research enterprise, which is at an all-time low. And there's a crisis in recruitment for clinical trials, and many clinical trials fail because of serious recruitment problems. When the citizenry is surveyed about their views of pharmaceutical companies and the pharmaceutical endeavors that are underway, only 14% of them say that it is an honest business, and that's right down there with tobacco companies and the used car industry. I think that we can probably do better. I don't think ads like this, touting participants as heroes, are likely to do as much good as the perception that the data that are generated from their participation will be put to good use now and in the future. And finally, it is essential to counter the cynicism that has arisen among doctors. And this study, which recently generated so much media attention, showed that studies of superior methodological rigor were discounted by physicians when they perceived that they had been funded by industry. Now, that earned a reproof from the editor of the journal, but many commentators have pointed out that doctors may have very good historical reasons to be skeptical. Whatever the reason, this distrust is very serious, because if doctors don't believe the evidence, what hope is there for evidence-based medicine? If they don't believe the clinical trials on which the guidelines are based, we can't expect them to act in ways that are based on evidence and that are consistent. So there are many problems. I'll mention just one. Where do we put all these individual patient data? What do we do with it? Where do we store them? Well, here's one solution that has arisen in response to this anticipated problem. This is a screenshot of Dryad, which is an online data repository where authors for BMJ Open can put their data, all of their data. And you'll hear today, I think, of many other inspired and inspiring examples of solutions to all the problems that that have been anticipated. Now, there are, of course, going to be problems we don't anticipate, but I believe those will be balanced by benefits that we don't anticipate. I have confidence in human and scientific ingenuity, and I think it is certainly the case we've only scratched the surface today in thinking about the benefits that might accrue from making clinical trial data more publicly accessible. Now, I don't think it's hyperbole to say that the matters we're discussing today mean that we're really engaged in one of the great struggles of human knowledge, and that is the struggle to liberate clinical trial information and make sure it is put to its best and highest use now and in the future. So it's a thrill to be part of this historic meeting, and I thank you for your attention.
We started a little early, so just in case if there are any immediate urgent questions on the current talk, we can take a minute or two. Okay. If not, we'll wait for the discussion period. And our next speaker is Dr. Robert Califf, who is the Vice Chancellor for Clinical and Translational Research and the Director of the Duke Translational Medical Institute. He's also editor-in-chief of the American Heart Journal. And before Dr. Califf starts, I just wanted to say, I mean, he has been one of the true champions of the sort of collaborative research and sharing out there in the world, and we're very honored to have him. Dr. Califf?
Great, thanks. It's a real thrill and honor to be here today. I think this is an important event in the history of human experimentation, and I hope that a lot of good will come from it downstream. I'm also sort of pent up here in this talk because I was told I could only say positive things about the benefits. Both of my sons are engineers, and I've learned the reason for that is that I'm attracted to problems, not to things that are going well. And engineers tend to think about everything as a problem that needs to be solved, and that's kind of the way I look at it. So, but I'm going to talk about the positive, and it's a bit of a personal journey, so I hope you'll bear with me. So the premise of all this, which I think has really been nicely reviewed by Elizabeth, is, at least from my view, trial participants give consent to be involved in a human experiment. I actually like to call it that because that's really what it is. The basis that enables ethics committees to approve this experiment is that there's a commitment to create generalizable knowledge. And so in a baseball analogy, I would say if the commitment is to create generalizable knowledge, a single is publishing the results. Unfortunately, even NIH-funded research, as Dr. Krumholtz pointed out recently in a review of clinicaltrials.gov, a very high percentage doesn't even get to first base. Hitting a double would be at least sharing summary data. Hitting a triple would be confidentially sharing detailed clinical data. And then there's a question out there lingering, very much raised by Elizabeth, would a home run be publicly sharing detailed clinical data? And if that were true, why would it be the case? At least in my experience, there are three really big reasons to share data across clinical trials. First is that trials often get results that seem conflicting. And, one of the sentinel moments for me was when the CEO of Pfizer, in the middle of the Vioxx controversy, came on the Ted Koppel show and said, you don't really need to worry about this out there in the public because your doctor will know what to do. If you think doctors can interpret conflicting clinical trial data based on looking at the data abstractly without any kind of expert synthesis of information, I think you're sadly mistaken. Demonstration or replication is a critical element of all of science, and I think it's equally true in human experimentation. But when the results truly do differ, it can either be chance or real, and only by multiple replications can you sort that out. Benefit-risk balance of treatment may vary as a function of patient characteristics, and in fact, we know that's the case. But knowing when it's true and when it's only a random play of chance really requires looking across clinical trials rather than simply within a single study. And it's increasingly a global issue that cost effectiveness is subject to the same issues. And as we look at new therapies like the new anticoagulants that we've just been working with, two of the big new ones, one on the market, one about to get there, entire countries are looking at it saying individually, this is effective treatment compared to warfarin, but can we afford it as a country? And if you can't afford it as a country, you got to make decisions based on the degree of benefit relative to risk and the cost involved. So it all seemed so simple when I started out because I was a coronary care unit doctor. When I started out, we prayed, we put people in bed for a couple of weeks, so we gave a little Lasix and morphine, and the mortality rate was well over 20%, more than 1 in 5. Along came the revolutionary knowledge that blood clots cause heart attacks and that we could open up these blood clots. And then a whole host of clinical trials ensued that I think set a paradigm, which to this day I still don't understand why it hasn't been followed by other fields. And the paradigm is pretty simple. In concept, you develop scientific ideas, you do the right clinical trials. If the right clinical trials are definitive, particularly multiple clinical trials, replicated, then it becomes knowledge, and the knowledge can be applied in practice and measured in practice through registries. At the middle of all this is measurement and education, which at its core is dependent on liberated data. At its core, it's dependent on many people being able to look at the data and draw the best conclusions. And so the way it happened, a lot of trials were done, not shown on here, the 90% of ideas that didn't work, that were weeded out because of the high requirement for data based on clinical outcomes. But those that did work required tens of thousands of people, not hundreds, to be really definitive so this could become a standard. And only a few made it through this high hurdle. But when they did, and we could measure whether the uptake was occurring, what we found was that when you've done it right and you've got definitive evidence and you apply the evidence in practice, there's a direct relationship to lowering the risk of being dead. Now, I'm not trying to suck up to Harlan here, but it made my day when his group published a study that showed the end result of all this was that your risk of being dead if you roll into a hospital in North America with your heart attack now is 40% lower than it was when we started. And I would argue this was not a matter of going from biology to marketing. This is a matter of a series of steps that were carefully planned and thought about by really smart people. Not me, mostly the people at Oxford and other people around the world. You invent and develop a technology, you do your trial, you add your data to the cumulative database, you generate new hypotheses, you do more trials, and you continue to refine. And that's really the way it worked in this field on a global scale. And to this day, as I say, I still don't understand why every field doesn't do it this way. So here's the end result of it. It started with the Antithrombotic Trialist Collaboration, individual patient data, 135,000 people up to 1997. This is ancient by today's standards, but still very important. And it was compelling when all the data were looked at that giving an aspirin was a beneficial thing to do. We then moved on to opening arteries for fibrinolytic therapy. And here there's another lesson to be learned. The data were entirely consistent across subgroups except for two. And you'll notice there the group with ST segment depression on the electrocardiogram, the risk-benefit calculation went the wrong direction, not statistically significantly the wrong direction. And people who came in late to the hospital beyond 12 hours, really not much of a benefit seen there. And the beauty of this is that multiple clinical trials showed the same thing. This enabled us, and we were privileged to be the coordinating center for the first AHRQ clinical practice guidelines. It enabled us to take a simple measure and tell doctors, look, do an electrocardiogram as quickly as you can it's an emergency, and if there's ST elevation, you better open the artery. If there's not ST elevation, it's a different scenario. And this led to a whole different set of trials in the non-ST elevation population. This was a result of combining data from multiple trials at the individual patient level to show that the absence of benefit in fibrinolytic therapy was reproducible in multiple clinical trials in that subgroup of the population. Not an isolated finding, but reproducible. But it was also the case that benefit diminished with time, and this was shown. Look at the numbers of patients here at different time intervals after arriving at the hospitals, and you see you begin to approach the level of no benefit. Well, we then moved on to percutaneous intervention, and people like me said, this is ridiculous. It's a technology that's so expensive, it could never replace the simplicity of giving a fibrinolytic drug. And it was hard to do the trials because you had to have a center that was capable technologically of doing the trial. It led to smaller trials being done, and it was really the replication across many trials that led to the definitive conclusion that on average, in most situations, if you have an ST elevation MI with a risk of dying of 10 to 25% within the next 24 to 72 hours, you want to get to a center that can do direct percutaneous intervention and open the artery definitively. It's better than fibrinolytic therapy. Many of us grudgingly accepted that knowledge, and it was really the replication across multiple trials that enabled that to happen. So this was the story of ST elevation, dramatic reduction in mortality. The key steps I've been through, it had to do not only with the inventions, it had to do with doing multiple trials, combining the data as a normal course. We didn't even really think there was much of a question. It was just what you did because of the way the Oxford people started it. And then you invent more therapies and do new trials. So one of the conclusions I came to this with as I was thinking about it is, this valley of death we're all talking about, there's a good reason that it's there. This path from biology to marketing is full of hazards. It's full of monsters that trip you up because most of what we invent doesn't actually work, or it has detrimental consequences that we didn't predict. And so my argument is that we shouldn't try to do away with the valley of death. What we need are really useful, intelligent guides to get across the valley of death, doing the right things as opposed to shortcut the right things. And I believe in the long run, trying to replicate the experience of the ST elevation MI global effort in every field by liberating data, making it available, stopping things that don't work more quickly, and reinforcing things that do work through the coordination of globally done multi-site clinical trials is the right way to do it. Let's go on to another problem dear to my heart, sudden death. Running a coronary care unit, we would see often people who died and got resuscitated. It's a tragic situation. You'd like to prevent it. And what could be more appealing than putting in something that if you have ventricular fibrillation gives you a shock internally? And sure enough, a bunch of clinical trials were done sequentially. And we had the privilege of working a lot with combining the data in this situation. And this just shows the time course of the different clinical trials. You had different circumstances, different enrollment criteria. But for the most part, one of the real beauties of this set of trials was that the baseline data and the outcomes were measured in a fairly consistent manner. And so overall, when we looked at the effect of ICDs in the clinical trials that were done, it's about a 30% reduction in the risk that you're going to be dead if you get an ICD compared to not if you met the entry criteria. A consistent result across multiple trials, except you'll notice two there. What was different about those trials? One looked at immediate post-MI patients, and the other looked at immediate post-bypass surgery patients. And so when you look at it from the point of view of hazard ratio, you see these two trials really stand out as being different. And I don't have the slides to show it all, but when you do the subgroup analyses and all the other trials and look at these same populations, this is a fairly consistent finding. And it's led to something you'll hear more about this year as CMS is enforcing the rule, but it led to a change in reimbursement where ICDs are heavily reinforced with really good reimbursement if you include the patients that are below the line. But in the first 30 days after MI or immediately after bypass surgery, you're not going to get paid. And in fact, they're currently, as I understand it, looking back at those who violated the payment rules and may take some money back from a few hospitals that violated that rule. But there was another problem that came up that was very controversial, where having data from multiple trials made a difference. In the initial trial that showed the benefit, made it 2, you notice the hazard ratios are similar between men and women, but because there were fewer women, the difference was not statistically significant. Now, most people who are knowledgeable about clinical trial data would say, so what? But there was a robust debate about whether women should get defibrillators, since there was not a significant benefit in women. And the second trial, major trial, Scud-Heft, really sealed the deal, because it also didn't show a difference. I'm sorry, this is really the outcome of ICD therapy, but it looked very similar to Made It 2, and when you combine the 2 trials, there was a significant difference with women. But then other subgroup characteristics turned out to be fairly consistent among multiple trials. Here's a simple parameter again, the ECG. If you have a narrow QRS interval on the ECG, you'll notice that there's a benefit, but it's relatively smaller. If you have a very wide QRS interval, there's a big benefit. And this is not a reversal of effect, detriment, and benefit. It's just a gradation in the degree of benefit, which leads to a very interesting argument about cost effectiveness of ICDs with a narrow QRS. And the initial trials only enroll patients with low ejection fraction, but later trials enrolled a broader spectrum of patients. And you can see that if you had a really markedly impaired left ventricle, very consistent result, except in the immediate post-MI period, of dramatic reduction in mortality. But in the group of over 30%, the reduction in mortality is less. And so this has led to a preferential treatment of patients with low ejection fraction, according to the CMS rule. Now, what about statins? One of my favorite topics. What could be more consistent than the effects of statins? But again, here's a case where data were liberated, and I think it's made a huge difference in the ability of people to make a statement about the class of drugs. It reduces everything that's bad for you in terms of cardiovascular events, no matter what you look at. But you look at the patient numbers here, they're really quite substantial. But a part of it that's really been a benefit of combining data, in my view, is to make the case, I think, beyond the shadow of a doubt, that we're not simply talking about lipid lowering as a concept. We're talking about the fact that statins, regardless of where your lipids are, reduce your risk of having adverse cardiovascular events. So if you look at this slide, you'll notice 45,000 patients in each group. From combining individual patient data, enabling the collaborators to look according to the specific groups of lipid levels, and you'll see in the second grouping there that even if your LDL cholesterol was less than 135, a very dramatic benefit there. And this is true at least down to 70, and there are trials going on right now to look even lower than that. Now, what about some emergency uses of combining data? I'm going to show you one of my favorite ones. We were in the middle of the Improve It trial looking at a drug called ezetimibe in lowering LDL cholesterol. The trial became under a shadow because of a controversy about a plaque regression trial that I think has been greatly misinterpreted, but nevertheless led to a controversy. We're in the midst of this very large trial, 18,000 patients being enrolled, ezetimibe versus placebo. We were limping along because of the controversy about the plaque regression trial, when a trial done entirely for a different reason, looking at aortic stenosis in Scandinavia, when they unblinded the trial, they found an excess of cancer in the group randomized to ezetimibe versus placebo. So here we are in the midst of an 18,000 patient trial. There's another trial that shows an excess of cancer done somewhere else for a different indication. And the question is, what do we do with our trial? Because the drug was already under scrutiny, many people said, this is it. You got to stop. But because we were working with the group in Oxford who were also doing a trial, we were able to, in midstream, without unblinding the principal investigators, with no involvement of the company in handling the data or the analysis. The company was involved in sending the results of the analysis to the regulatory groups around the world, which is their obligation. By the way, the New England Journal completely misunderstood that in an editorial that they put out about it. But the fact is, we were able in midstream in ongoing clinical trials, our data monitoring committees, and Richard Pito at Oxford were able to put the data together from these ongoing trials and come out with a statement. And this took individual patient data because, remember, one of the key issues in cancer is the latency period between the time that you're enrolled in the trial and the onset of the cancer. We're able to show beyond the shadow of a reasonable doubt that when you combine the data from the two big trials ongoing, that the result from the trial in Scandinavia was a fluke. Absolutely not a hint of an excess of cancer, and not a hint of an excess of cancer at any site. So this shows that when you handle data correctly, and when it's handled independently of the companies by coordinating centers who know what they're doing, it's possible on the fly in the course of 2 weeks to combine data from ongoing trials and produce relevant data for regulators that overturns play of chance findings in single individual trials that are ongoing. So we concluded from this that we should continue our trials, and one day we'll finish the IMPROVE-IT trial. It's still ongoing. Then the last example I want to give is one where disregard of liberation of data, disregard of combining data, I think led to terrible adverse consequences for the American public. What can seem more obvious than if you have renal disease and you're anemic, that you should get erythropoietin and get as much erythropoietin as you need to, quote, normalize the hematocrit? It seemed very obvious, and it was so obvious that our single-payer dialysis system made it a clinical practice guideline. This was done brilliantly by dialysis companies and American physicians. They gave a lot of high-dose erythropoietin. Problem was they forgot to do the clinical trials. They skipped from biology to marketing. And what happened was one of our fellows said, why don't we do a trial on people not yet on dialysis, but who may need dialysis soon because they also do worse the more anemic they are. This was a choir trial, which we did. Unfortunately, the trial was stopped in midstream because the group randomized to the higher dose of erythropoietin was doing worse, not better. But the trial was poo-pooed by almost everyone because they said it wasn't a well-done trial, it's probably not right. When the fact is, when all the data came in from all the trials being done, high-dose erythropoietin was bad. And you can see from the way in cumulative meta-analysis, the way it lines up, that this could have been detected much earlier, and it could have been detected much, much, much earlier if the right trials had been done and the data had been combined. And then the very final example, to show that this can cause perplexing things to happen that you don't understand. Here's beta blockers for heart failure. What could be more obvious? It reduces your risk of death, period. Definitive results with huge treatment effects. But look at all these trials when broken down to U.S. and rest of world. Now, I'm not here to argue that beta blockers don't work in the U.S., but when 5 out of 5 trials show that the effect in the U.S. is much less than the rest of the world, you've got to begin to believe there's something going on here, whether it's other treatments, genetics, or what, I don't know. But this is an unsolved mystery from combining data. So in conclusion, I would argue that liberating data across clinical trials is ethically sound. It clarifies findings for policy recommendations. I tried to show you that. It often raises new questions. And when this evidence step is skipped, as it was with high-dose erythropoietin, there's a significant hazard to the public health. One element of this evidence step should be, I believe, the sharing of patient-level data across trials. But since I'm not allowed to say anything negative, I'll just close with a point. There are a lot of details to work out, which I'm sure will be completely worked out during the course of the next day and a half. Thank you.
There will be time for negative comments, of course,
Tomorrow at 5.
as we go into the next discussion. So I think we picked up some time, which is great because I think we can use the discussion section to include everybody. But right now, I'd like to. I don't know. Did we have signs? How are we doing? Are we bringing signs up? Yes, that's good. So why don't you bring the signs up, and I'll introduce all three of our discussants at this point. And Basically, just the way that we have organized this is to ask each of our discussants to spend maybe up to 10 minutes doing a non-slide presentation to comment on aspects of the two opening talks and the general themes. Our panelists are probably many of them are well known to you. There's Dr. Harlan Krumholtz, who is the Harold Hines Professor of Medicine at Yale and has been very, very active in the field, as Hen already referred to, in terms of what might we do with data sharing and learning about various things like cardiovascular risk factors. Miles Axton is the editor of Nature Genetics. He's actually a true molecular basic scientist, so perhaps the broader theme, although we're talking about clinical trials, data sharing has a wider application. And lastly, Dr. Jesse Berlin, who was at the University of Pennsylvania and then joined Janssen & Janssen as the vice president for epidemiology. So, Dr. Krumholtz, perhaps you could lead off with some comments? Okay. Thank you.
I'm so pleased to be here today. I usually speak extemporaneously, but I wanted to get my thoughts down more formally to share with you. This conference was a dream of mine, and to see such a diverse group of individuals here to discuss the topic of open science is nothing short of remarkable. I want to apologize, though, that I can't stay for the entire conference. When I was invited, I'd already committed to speak at my public library in my small town where I live, and although the workshop represents a major step forward in the dialogue about open science, the flyers were out and I didn't have the heart to say no to the librarians. They're a very earnest group. But tonight, when I'll be speaking with a small group of people from my town about healthcare decisions that they face, I'll be thinking about the conference, and I hope also that you will be thinking about people like that as you discuss the issues today, because this conference really is only about the people who are every day facing those decisions. Open science can have many different meanings, but for that reason, I want to focus your attention particularly on these people who face the healthcare decisions every day and depend on the availability of all the information that's relevant to the options that they are considering. So we can talk about increasing the efficiency of basic science. We can talk about sharing of control data. We can talk about how we make things better in terms of networking and sharing. But, and I think these are vitally important to the generation of knowledge that's going to help new innovation. But I would like us particularly to focus intently on the sharing of data that are relevant to decisions people are making today and that they face every day, recognizing that the missing data is impairing their ability to make informed decisions, is essentially keeping from public view information which is vital to true informed decision making and has so far hampered much of that. Our experience to date is that every time data are shared, voluntarily or not, we learn something new and important. It happens time and time again. In every case I know when data were released, especially participant-level data, the assessments of the risks and benefits of the drugs and devices changes in important ways. We learn something new. Granted that many of these episodes were in the context of concerns about products, such as Vioxx or Avandia. But still, I believe it's likely that we will learn much new information from data sharing. And in many cases, I believe that it will build in a complementary fashion to what is known. It doesn't necessarily have to be understood as a polarized situation where we're sharing data in order to oppose information that's out there. It can strengthen and help provide nuance and understanding. As Elizabeth said, important study, I believe, that they published in BMJ suggested that unreleased data in many cases may strengthen evidence for a product. In fact, may as often strengthen that evidence for a product as it weakens it. In any case, what's important is that we support the idea that data are a social good and the best science takes place in the light. In that spirit, I have a special message for our industry colleagues who are here today. Your presence here is important, and I want to say very deeply appreciated. We know together that resistance to sharing data is not an issue that exclusively resides within industry. Let's just admit it. In fact, our group, led by Joe Ross, has published evidence that even the NIH-sponsored trials often, commonly, at a crazy rate are not published. That, maybe even 3 years out, a third of them are not published. Now, I will tell you, the NIH has taken this seriously. Last week, they had a meeting. Francis Collins convened with the institute heads, and they're thinking very seriously about how to address this problem. So we know, let's acknowledge it. This is not an industry issue exclusively. That being said, those of you from industry know that industry has a special place in society and a special opportunity for leadership due to your organization, the way in which you own the data and control it, the kind of actions you can take. And I believe you have a meaningful motivation. The profession has less trust in your science than in NIH-sponsored studies and is less likely to act on the results of the trials you sponsor, not just the ones you conduct. The pharmaceutical and device industries no longer have the respect they once held. We know the events that contributed to these negative perceptions, and there's no reason to go into them today. But the result is a situation that does a disservice to the public, the medical profession, and the vast majority of professionals in industry who have extraordinarily high integrity and are in that industry for the right reasons. It's time to restore trust and reclaim your position. Industry is replete with those of you who have, I believe, have the highest ethical standards. You were drawn to work in industry because of your desire to contribute. And you know how much we depend on industry for research funding, for innovation, new ideas. It's the way we bring products to market. It is the way that we help people through that collaboration and partnership. Making meaningful actions to share data will be a big step in the direction that will help us restore and put industry back where it belongs, as a trusted source of information, as a place where people look to for help and understand as being full of individuals who are dedicated to that. There are already many heroes in industry and evidence of progress. I've heard from so many of you who are committed to this path. I want to particularly acknowledge Richard Kuntz. He isn't here yet, Chief Science Officer at Medtronic. You'll hear about this project later, but he engineered a groundbreaking decision to release fully the company's data on a product that's nearly $1 billion in annual revenue. Medtronic made clear they were seeking the truth, were willing to have others review the data, even while their own scientists were doing the same, without restriction. I spoke with John Waldstreicher last week with Jeff Nye, and she conveyed to me Janssen's commitment to share data and provide leadership. I heard news from Frank Rockhold, another hero, about remarkable changes in practice at GSK, where they are committed to releasing participant-level data and allowing the free flow of information that's going to change the game. I'm hopeful that others will follow. These individuals are not just talking about sharing control group data, as I said, or information on discontinued drugs, but sharing meaningful data relevant to drugs and devices that are on the market. They realize the studies are only possible through the generosity of people who consented to participate, and that we have an obligation to ensure the efforts of those subjects contribute as much as possible to knowledge generation. And by the way, that's going to be essential to ensure the continuing flow of individuals who are willing to participate in our trials. They also realize that in return for the privilege of selling a medical product to the public, industry bears a responsibility to ensure that all the data concerning the risks and benefits are available for everyone. I'd like to invoke a quote from Peter. I'm almost done. That he delivered in the European Parliament in Brussels at a conference on the common good. He said, "If commercial or academic success depends on withholding data that are important for rational decision-making by physicians, patients, and governments, then there's something fundamentally wrong with our priorities in healthcare." I couldn't agree more. So let's get past whether it should be done and have the conference focus on how it can be done well and responsibly, attentive to the needs and concerns of everyone who's at the table and wants to participate in this discussion and has a stake in what we do. We must minimize the possibility of abuse, and Rob's shown that there is some possibility of abuse, and maximize the probability that sharing will make research more efficient and effective, and ultimately support better decisions by patients. We need not fear data sharing. Science is self-correcting. Let the public dialogue and the scientists debate. And the public dialogue can then progress if the data are available. And let's be clear about what's needed. We need to have studies published and summary results reported in clinicaltrials.gov. But that's not enough. We need to have individual patient-level data broadly available for investigators, and not parsed out to selected investigators, but available broadly and freely. We need the protocols and case report forms. We need full sharing of the source data. If we have the will, we just need to find the way. And with the talent in the room, and with those listening on the webinar, and those who are interested, I know solutions can be found. If we're committed to the path, we can figure out how to do it. I see a day when we're all truly worthy of public's trust, and that we recognize our obligation to patients by ensuring that we are open with our science, including academics. Point at me. My own profession is not good at this. Mike has pointed this out to me. You're absolutely right. We are not good at it in academics. We need to fix this. I see a day when companies are competing on science, not marketing. Companies are competing on science, open science, not marketing. And open science and data sharing are part of their business model. I also see a day when academics get credit, not only for the papers they publish, but also for the papers that are published and the knowledge that is generated from the databases that they create and share. That credit comes to them from what others do because of what they made possible. I see a day when it's in everyone's best interest and is expected that data sharing is an essential characteristic of being a good scientist and a good citizen. When I look at the audience in the library tonight, I'll be very proud that we are taking steps to ensure that in the future their decisions will be more informed because of the free flow of information. And they will feel more confident about academics and industry as they see us working hard together to ensure data relevant to clinical decisions are rapidly shared and openly discussed. And the together is an important part to me. That is, we are finding ways to open dialogue, to have mutual respect, to work constructively, and to seek ways that we can find answers together, recognizing that from the different perspectives, there's a reason why we have the current system. And it's going to take some sensitivity, some listening to each other, to working together, but keeping our eye on what is a societal good in order to make the progress. But those people in the room tonight, when I'm talking to them, they should be impatient with us. They should be impatient to see us make real progress, get past whether to do this and solve the how, and advance forward quickly. So let's get moving. Let's show the public what we can do together. Thank you.
Dr. Axon, please.
Thank you. I'm delighted to be invited to speak. Nature is very fierce about access to methods, data, and materials. Papers that are published in Nature research journals have to show they're working. That's important for the integrity of the science, and it's also important for the impact. Good citizenship is also good business. In other words, your results matter to others, and your reputation is made in other people's labs. So when you make data available through whatever mechanism is available to you, you are promoting your own research and the value of your research to other researchers and to society. I've been involved in quite a lot of experimental attempts to allow access to research data, including publishing community papers on the database of genotypes and phenotypes, the Genetic Association Information Network launch, microattribution as a way to incentivize community annotation of the human genome. I've editorialized about the portable legal consent for common genomics research. I'm undertaking an experiment with peer review on an open commons platform in order to see whether fully open data sets can be used in multiple research projects. But I think what I would like to talk about today is what is the most pragmatic way to immediately move to a way to share clinical trial data in a way that other trials can benefit from it, but also we can get quick knowledge turns in terms of hypothesis generation, validation, and new hypotheses for basic research. It's that flexibility in reuse that is afforded by the individual level data availability that is important. So what can we do? Obviously, the funders would love it if all the data funded by public funding could be put in a public repository, and everybody could, who is a qualified researcher, could come in and see and use that data. I think the idea of a repository, either at the industry level at your own institution or your company, or in a public place, in the case of publicly funded science, I think is a very good idea. But what I don't want to see is the separation of the people who have invested their time in the trial from the data. And I think there are two reasons for that. One is the knowledge that comes with the data. And I see two tracks, essentially, for reuse of data. One is reuse of the data with the original trial organizers as co-authors, in which case they're engaged in the analysis and it's a formal collaboration. The other track that I see is that the data gatherers, the trial organizers, are cited as collaborators but not authors. And that allows the original trial organizers to distance themselves from the conclusions of the reusers of the data. And I think that's very important in order to get some independent analysis, but it's also important that the people who spent their many years organizing the trial still get credit in the publication sphere. Even if the conclusions end up being critical to the trial, I think it's very important to keep that provenance there. I don't like the idea that data can be removed from the people who generated it, and I don't think it should be removed from the people who participated in the research either. So I'd like to say something a little bit about the research participants. Now, opening up individual level data to research participants can generate important insights that can improve trial quality, recruitment, utility, and research use. I think because of the original consent agreements that are in place, the research subjects engaged in a trial should not attempt to identify their own or others' results, just as researchers are prevented from doing that, unless that's part of the original clinical trial design and that they're actually engaged in commenting on their individual results. Because I think we need, if we're going to move to a data sharing mechanism, we need to be able to encourage people to participate in research. And that means we need to, not only the legal requirements that are in place, but also the encouragement of future participation of research depends on handling this properly. But I do think because research participants are spending many more thousands of hours engaged with the particular problems than are the researchers, there is a unique perspective that we can gather there. And so I think it's very important that we think not only about qualified researcher access, but also research participant access to results. I've done some experiments with how you would obtain access to research results, and I think it's essential that anyone wanting to reuse the results put in a request documenting status as a bona fide researcher with a research plan detailing aims and objectives of the research to be performed with the requested data, specifying the data set necessary and sufficient to conduct the proposed research, and an analytical justification for the research data set, and a detailed documentation on the processes to ensure that the data will not be distributed to third parties, will be secured and protected to safeguard the privacy of the research subjects. The default, I believe, should be that access should be rapidly granted by the trial organizers and owners of the data. If not, I think a data access moderation committee model is a good one. It should include members of the trial group and independent researchers and or participant representatives. And they are responsible for advising those who've been denied access how to comply with conditions for access. This model may be quicker than the NIH DAC procedure and may work better since it's envisioned that the trial group will remain involved in data reuse analyses and in publications. And I went through the ICMJE guidelines and the Medline guidelines for authorship, and it seems that we already have in place the correct attribution for authors and collaborators. So I think those are in place. We have a model for a data access moderation committee with an industry group that generated confidential HIV data using NIH funding, made it available to researchers, and we have that document available. Journals. In the case of policy-ready science, I think we have a responsibility to publish any dissenting analyses by the data provider groups in the event that data reuse generates differing analytical interpretations, provided these pass normal peer review and journals quality filters. And in the case of policy-ready science, the journal has a responsibility to assign peer reviewers who are familiar with the needs of policymakers and to explain the appropriate societal perspective and the implications of the results published. So I think we have a model where we could pragmatically go immediately to full data sharing, but I don't think we should remove the original data generators and the research participants from that process. I think they need to be given credit using existing mechanisms that we have, and I think journals have a part to play in that process. Thank you.
And before we open up the floor, Dr. Berlin.
All right, thank you. Good morning, everyone. I'm excited to be here and very grateful. I'm hearkening back to my days in my PhD, or SCD actually, which was on the topic of publication bias. So not publishing results based on lack of statistical significance. So this has been kind of a passion for me for quite a while. So I want to start by asking the question, do we always need participant level data? And just as sort of a heads up, the message here is let's be careful about when we really do or don't need person level data. And so if all we're doing is asking this kind of overall question about treatment effect in general, then we're going to get the same answer whether we use participant level data or individual or summary level data, right? Just for this overall question, we're not getting back in and re-adjudicating events. Just everything is exactly the same, then statistically we're going to get the same answer. So that's just kind of a little bit of background from the statistician that I used to be. But the point I think that's been made a couple of times this morning is that the data are not always the same. That when you get to the participant level, you may decisions may have been made early on that you wouldn't replicate. So published studies may not, for example, have always done the appropriate intent to treat analysis in a clinical trial. Sometimes you want to do a proper time to event kind of survival analysis, and you can't often do that just from published data where all you have is a count of events and a numerator and a denominator. And then I think importantly, as has been noted already, is getting into this question of adjudication of events. So are the endpoints defined the same way in the different trials? Were they properly validated? So what I want to do is highlight some points that were really already made by Drs. Califf and Loader, particularly focusing on this idea of exploration of heterogeneity. And what do I mean by that? We know that for a given clinical question, that the study designs, the specifics of study designs and the patient populations, certain aspects of how the treatment was administered vary from trial to trial. And we also commonly see that results of estimates of treatment effect vary from trial to trial. And the goal then becomes to try to understand where are these differences coming from. Is it some function of how the trials were done? Is it some function of the subgroups that were included in the trials? What's going on? So we get into this question of exploring predictors of treatment effects. So now this can be done at the aggregate level. So if all we have are the summary level data, then we can do things like if we want to know whether treatment works better in men than in women, we can look at the percent of males in the trial at this aggregate level and kind of examine the relationship between the percent of males and the treatment effect. Now, it turns out that this is an example of what's called ecological bias. And the message I want to deliver, I'll give you an example, is that kind of approach can go seriously wrong. So it's not just a matter of feeling more comfortable having the individual level data. You can actually get wrong answers. And there are also issues that I think Dr. Zarin mentioned in terms of statistical power. So the example I want to talk about is look, it's an old one. It was from my days as an academic before I started wearing my Darth Vader suit wherever I go. So this is looking at anti-lymphocyte induction therapy in kidney transplant patients. So this is kind of prophylactic anti-rejection therapy. So instead of waiting for the kidney to get rejected, you give the therapy on the same day as you do the surgery, right? We had done an aggregate level analysis, and then Linda Sezek, who was a fellow at the time, always nice to have a fellow to go out and do the groundwork. So actually contacted the 5 authors of the original studies and managed to get the person level data, the participant level data. One guy actually had the data in punch cards in his garage. So I think we've come beyond that. So the factor I want to just mention, which was the point of the publication that we got, was this panel reactive antibodies. These are a measure of immune system sensitization. So high level means you're sensitized and you're more likely to reject the kidney. And the message here, the bottom line kind of is that if you try to look at this at the aggregate level, you get confusing, inconsistent results. The results at 2 years differ from the results at 5 years. It's just not clear what's going on. When you get the patient level data, the participant level data, then you can do an appropriate analysis and find out that in fact treatment is extremely effective. So meaning like an 80% reduction in risk of rejection of the graft. So an 80% rejection in the people who are sensitized and no benefit in the people who are not sensitized. Okay, so that's an important thing to learn because it's only in the trials that the sensitized population was only about 15% of those who were treated. And these are strong drugs. They have consequences, unintended consequences, like lymphoma, for example. So you really want to try to focus the treatment on the appropriate population that's going to get the benefit and not the risk. And again, I think it's the key here is that that's something that we would have missed if all we had were the aggregate level data. And actually, to Rob Califf's point, what's interesting is you can look at these studies individually now and see this difference between subgroups consistently replicated across all of the studies. So it's not just an artifact of one of the studies. Another area that I want to highlight is the use of participant-level data in addressing safety questions. And I want to thank Rob for mentioning the erythropoietins. I'm actually going to mention an example that we participated in the oncology side, so not in the kidney disease. So this was in around 2008. One of the Cochrane groups came to the 3 companies, us, Amgen, and Roche, who make these erythropoietins, to look at, with a request for patient-level data to examine the mortality question with the erythropoietins in the oncology setting. these drugs in oncology are used for chemotherapy-induced anemia. So the findings are published in The Lancet. The advantage in this case of having the patient-level data was if all you have are aggregate level data, you can look at things like what was the requirement for hemoglobin level for entry into the trial. What was the target hemoglobin? So was it a high target or a low target? So you can look at those trial level characteristics, but you can't get down into the details of what was the individual person's hemoglobin level at baseline. So there's a lot you can do with the patient level data that, again, you couldn't do just from the aggregate level. And you do get different looking results depending on how you do the analysis. The advantages of adjudication were mentioned. I'll just throw in another example that with the people will be familiar with the SSRI antidepressants, where the finding was that there's an increased risk of suicidality in adolescents and young adults. So that came from starting with the patient-level data, where you had to get in and actually read the narratives and decide. And in the original analysis, FDA had outside experts, completely independent from FDA or from the companies, getting into the data and adjudicating these individual stories and deciding, was this a suicide attempt or a suicidal ideation, et cetera. And that ended up in a label change for those drugs. So I just want to raise one other question of whether there might be an intermediate step here. And this has been discussed some in the literature, is the idea of sharing clinical study reports. And these are much more detailed reports than you typically find in a publication, even an online publication where some of the data are provided in electronically. So this is the detailed analyses, the subgroup analyses, the sensitivity analyses. Did we pick the right statistical method? I'm out of time. I'll talk fast. So just as a kind of intermediate, quick, kind of low-hanging fruit, as they say, I want to raise this possibility for further discussion of whether we might at least gain some ground by disclosing the full clinical study reports. Because in addressing some of these subgroup questions, you'd actually get the data that you need from the clinical study report that you wouldn't typically get from a publication. And I think I had some comments about how do we do this carefully, and I think that's really the topic for the next day and a half. Just based on the experience that we've had, there are a lot of details, starting with just harmonizing data sets. I mean, you can't underestimate, you can't overestimate, sorry. It's a lot of work to get things, you're taking data from 3 companies, from studies that are 20 years old, and turning that into an analyzable data set was the first 90% of the work. And the 10% is somebody doing the statistics. So there are some non-trivial, and just how do you set up the dialogue between the data providers and the people doing the secondary analyses? How do you ensure that there's appropriate contribution to the science without undue influence? And I think I'll stop there. Thank you.
Perhaps we can have Dr. Loader and Dr. Califf rejoin us because we're going to open the session to the floor now for questions. I think Dr. Zara might have a lead general question for the group.
Actually, I was going to say I will save my question. Let the conversation evolve, and if we have a lull, I'll throw in a question or two.
Okay. Well, actually, before anyone gets up to the mic, we already have a question from the floor from Diane Paul of the as a patient advocate, specifically to the panel, but directly to Dr. Axson. In instances where patients don't improve while in trials, isn't there an obligation to allow patients to know whether they got the experimental therapy, since it may affect their future care if the agent is available off-label or approved in the future? So isn't there an obligation to allow patients to know whether they got the experimental therapy?
I'm the last person to answer this. I have never run a clinical trial, so I have to say that I'm going to go with the original consent, because what I said here is that we must stay with the original consent, and part of that consent process is explaining the possible outcomes of the trial to the participant at the outset. That process, I think, is absolutely essential to stick with it. Now, there may be cases in which the trial organizers just take a look at the results and they say, okay, we really must, for the good of the patients, unblind this trial. But I think that is not something that a journal would force or a funder would force. That is entirely an ethical issue for the licensed and qualified physicians who are actually running that trial and caring for the patients.
I would also add that the phrase that people use in that area is returning research results to the participant. And in this country, IRBs and the investigators, well, the IRBs oversee that process, and there are guidelines for when that's appropriate, which is extremely important, but somewhat different than what we're talking about here, which is sharing data more publicly, sharing de-identified participant-level data for the purpose of further research.
I mean, please comment. No, it's open to the panel.
Yeah, just a quick comment. Many of the trials we do, and there are pluses and minuses to this, but in many trials, people complete the randomized portion of the study and then are allowed to cross over onto the active therapy. So the problem is that it then becomes a single-arm study with no comparison group. And so anything that happens, whether it's good or bad, kind of tends to get attributed to the therapy, and that may or may not be right.
Yeah, I would just quickly comment that it's often done, and as Deborah said, there are certain issues that come up. For example, if there is a continued long-term follow-up, there may be reasons not to reveal the treatment, but that requires a separate consent when that is done, so that people know it. But I also think there should be an obligation to inform people about the vagaries of knowing what treatment you are on relative to whether you actually benefited. This is not a simple issue of knowing that you benefited because you got the active treatment and you felt better, for example, because that could have easily been a placebo effect. So there's a certain amount of discussion that's actually quite startlingly interesting when you reveal results, particularly to a group of people who have participated. The women's health studies with HRT were probably the best example of that in my career, where it was a devastatingly adverse or neutral, depending on how you look at it, impact, leading to really interesting discussions about what it all meant.
I mean, for the benefit of the individual who, asked the very common sense question, why, if you participate in a study, why don't I get to know what I was on? I think what we're hearing here, and it's important to make the distinction, is that this is a very complex issue, which is almost the subject of a different discussion, because it has to do with the unintended consequences of revealing information that might not really be informative, as Dr. Califf just said, not to mention what's in the informed consent, but that the topic here really is not addressing identified, not linking data to individual patients. So I just wanted to make that point very clear. I think what we're talking about right now intentionally avoids going into that space. But the other question is a very good one, I think, for a different setting. Thank you.
My name is Cindy Gagan, and I'm a patient and an advocate, and I am an advocate for this data sharing. So I appreciate being here, and I appreciate you convening this meeting. As an advocate, one of the things we need to do is be able to explain the benefits of this to the public. And as I think it was Diane Paul's question is what we're basically asking is for participants to be content with summary level data. So as a participant who's invested in the clinical trial, now my case reports, et cetera, are de-identified and not connected to me, but they're going to be shared broadly. But I am the participant asking for a participant level data. And I'm not saying I understand the reasons for it, but it's still going to be a hard bridge necessarily to cross, especially when you come to things like incidental findings, not necessarily which drug I took, but you might find something else compelling in there, and it's just another issue I think maybe cross later. But if we're asking the public to accept summary level data, and then we're trying to benefit people by participant level data, we should be sharing the participant level data at the participant level, perhaps.
So let me take a first stab at this. I think that whether or not the participants in the clinical trial receive information after the trial about their own experience is separate from whether or not the participant level data in a de-identified way is made available to other researchers. So even if in a trial, or even if policymakers decided that participant level data was never made available publicly for research, it doesn't mean that the IRB decision to whether or not you as a participant got the data, it's just two separate things. So each trial would have its own guidelines about, and your informed consent process would have said what you would receive, and it's just a very separate thing from whether researchers have access to that information.
I'm agreeing with you, and what I said was that the research participants should have a right to the individual level data. I think that's a very important distinction from what the doctors are saying. I think that's a very important issue, but what I asked was that the individual research participants not try to identify themselves or anybody else in that trial. Now that may be pretty annoying to you if you're trying to make decisions about your life, but the clinical trial is not there for you to make decisions about your life. That is your treatment and your physician interaction. I think as part of the research activity, research participants can recognize patients like themselves within the clinical trial data and can make observations about the individuals involved that may actually either lead to research or be in themselves research activities. But I think you're engaging the individual level data with your unique perspective as a researcher.
Yeah, well, I mean, please comment more, because as we hear this, both the opening question and yours, Madam, do strike one is very important, because one can easily appreciate that from an advocate perspective or patient perspective, this does seem like a sort of almost arcane distinction that we make up here. And I think what's on the table is how do we communicate the consistency of how do we communicate the goals properly here? So I think we should continue this, because I think this question is important and we hadn't really registered until we heard it. So maybe Dr. Loader first.
I will just be brief, but the matter that's raised is in some ways separate from what we're discussing, but in other ways not, because certainly when I began as an investigator in clinical trials, it was not standard practice to return to the individual investigators in the sites their own data. In other words, we did not necessarily ever learn which of our enrolled subjects had been on treatment or not. At least it was not standard to return that information. I'm happy to say that at least in academic medical centers, that's rarely now the case, because we're not allowed to sign trial agreements that don't involve some level of access to the data. But I think that the lack of a sharing culture, especially earlier in clinical research, in part led to participants never understanding what happened to them.
Well, I want to thank you for that comment and say that if our goal is in part to restore trust and to build these relationships, then I think we first really have to listen to what people out there have to say. And if we're just talking today about how researchers can access data, it will seem as if we're self-serving, even as we're trying to promote society's best interest. And maybe it's true that we ought to think about the idea if the data are a societal good, and if we are indebted to those people who have participated, what's our obligation to them? And giving them a dense consent form that they've signed because they trust us. I mean, they don't really necessarily, let's be honest, understand deeply every line of that consent form, but they trust us that, one, we're going to make best use of the data, and that's what the primary purpose of this discussion is, how we can fully harvest the knowledge that can be generated from their generosity. But the other thing is, what are we going to give back to them? And maybe we should adopt some principles around that. I felt in the trial that we ran, I just thought we didn't think enough about how we could make sure that everyone who participated in that trial knew what the result of the trial was and what that implication was for society, let alone for them as an individual. It was so easy to have them participate in the trial, to take their data and to call the trial over at the end and just say thank you mostly to the sites, and not have an individual interaction with the participants and making sure that they understood what had accrued as a result. Similarly, if those participants are interested in working with others to see what's going to happen next, I mean, Sharon's working so much in citizen science. I mean, these data sets are going to be available. They need to know what opportunities there might be if they're truly interested in this topic. Anyway, my message, I guess, is that if one of our goals, academics, industry, everyone who's at stakeholders here, is to show that we are listening and that we want to participate, then we have to be sensitive to that really key comment that you're making in the kind of way in which it will be reflected by society as a whole when they hear us. I could see easily for them to say a bunch of people got together in a small room, talk about how they were going to share data with each other and enrich themselves with opportunities and what's in it for us. And yeah, we hear that they want to do more research for us, but I'm not seeing the connection. So there may need to be a part here that pays a lot of attention to that also. And thank you for that.
I feel like I'd like to clarify a little bit about what I said, because I think it actually is clear and there should be a standard, and it would be fairly easy to articulate it. I mean, in general, there should be an obligation to inform participants in clinical trials about the result of the trial. And 95% of the time, and in fact this is a standard because the blinding assignments in blinded trials are returned to the sites, 95% of the time it's perfectly right to inform someone what treatment they got, placebo, active treatment, or if there are multiple active treatments, which one and when they got it. The only point I was making is when you do that, you have a second obligation, I think, to do your best to make sure that the participant can put their individual assignment in the context of what a clinical trial is. And that will take some time. The exceptions, I think, are fairly clear. It's when you have a follow-on period to the trial where maintaining the blind, for example, is critical to the scientific purpose, but that requires a reconsent. Otherwise, I think it's a fairly simple standard that ought to be. Yeah, and I just bet if you audited all the trials, it's more an opt-in than an opt-out. I mean, if people are persistent and come to us, we will tell them, but have we really tried to provide it as a service and make sure that it's easy and that we provide the support you're talking about? It's not just saying, here's what it is, but are we building that kind of bridge? Usually we're done with that trial, and we're moving on, and if someone's persistent, we'll do it. And I think you're right, Rob. Maybe it just would be nice to think about that as articulating that standard and trying to talk about what best practice is and trying to make sure we're adhering to it.
Yeah, actually, we'll move on to another question, but while we were discussing this, more questions came in on the same topic, asking the same thing. So I think what we're hearing, it's very important that we clarify a standard here, expectation, and what can participants expect as well. And I think we'll take that to heart and follow up on that. Thank you. Yes, sir.
Peter, is it on? Peter Doshi, and I guess, sorry to shift back, but as from the perspective of a researcher, and I liked how the discussion already sort of, Dr. Krumholz talked about moving from whether to share data, but how, and Dr. Berlin immediately responded with a low-hanging fruit of sharing clinical study reports. And, in the course pack for this meeting, I have an article, The Imperative to Share Clinical Study Reports, so I was really happy to hear that. Now, my question is whether we could flesh out, at least in part, because I know time is limited, the details of what that would mean. So, for example, I know that clinical study reports are written to a pretty strict set of guidelines, the E3 document put out by the ICH. And part of the structure of this is to include as an appendix the individual participant data. This is now a printed out PDF version of individual participant data. And so if I have all that data, I really wouldn't know what to do with it, because first I'd have to scan it and then try to convert it back to a database that it originally came from. So it would seem to make sense to bring in the electronic data set as is. So I was wondering if that would be part of the proposal. So I guess the general question is that specifically part of the proposal, and what parts of the CSR are in that offer of the low-hanging fruit? And the other question is this idea to offer clinical study reports to everybody, or is this going to be on a case-by-case basis? The article in the course pack describes our inability to obtain clinical study reports, full clinical study reports from Roche, despite their promise to make them available. Thank you.
So the first part of your question was that about the individual patient data, and that's not what we're proposing at the moment. I mean, we're certainly here because we're interested in figuring out the best way to do that. That's not what we're proposing initially. So again, just to be clear, I'm not saying we're not going to disclose individual level data, but that's not what we're talking about when we say clinical study reports. And there's also a lot of stuff in these reports that's, let's say, when some of my FDA friends talk about don't editorialize, just give us the data. So there tends to be some editorializing in some of these. So we're really trying to think of a way to share the clinical study report, the data piece. Of the
just to be clear, are you speaking for Janssen? No, I'm just wondering if you're, when you say we, or, I mean, I just mean, do you mean you're talking about your own personal commitment? That I just want to be clear, that's all.
So we're early in our discussions within Janssen. So none of this is official Janssen position yet. This is what we're striving for. This is what we're working toward. We've had some early discussions at some fairly high levels. I don't know if Joanne Waldstreicher wants to add anything to that.
Yeah, I mean, just to add, we are moving towards greater transparency, and we, as a first shot, we are in favor, and we'd like to collaborate to make individual patient data and clinical study report data available. The status that we are in right now with everybody else is because of consent forms, because of information in our clinical study reports. For example, individual patient narratives are in there. So we're here, we're at the table, we want to work together to figure out how to make this happen together, and we're very much in favor of moving forward.
I mean,
I wasn't saying that in an accusatory way. I was just trying to understand if you were speaking on a policy point of view, that's all.
So just a quick follow-up, because I'm not sure that the original question, people fully understood that the clinical study case reforms to which Dr. Berlin is referring would not include, obviously, the participant level data, right? So, I mean, just to be very, very clear here.
Right. That's as a first step. That's what we're saying.
So can you help? I was going to say, could you help us understand? Maybe there can be a little discussion. What additional would really be gained by making that available? I'm not sure everybody would understand. Well, what's really different from the publications? You've sort of said there is difference, but I'm not sure people have a real sense of why that's different.
I want to make sure and comment on that too after Jeff.
Okay, I should just defer to Rob, who's actually much more experienced at this than I am. I mean, typically what these reports have is a much greater level of detail about methodology. So all of the minute details that might matter to interpreting the results are in the clinical study report. The sensitivity analysis, so we chose a particular statistical approach, but here are the 5 other approaches that might be applied. Here's the detailed table of all the subgroup reports. So if I'm interested in these subgroup effects, I can get those from aggregate level data within the subgroups without needing the individual patient data. So that's really where I'm going with it.
And I just want one point of clarification, not in addition to this, but so I just want to be clear what I'm hearing from Janssen, because Joanne, this is, I think, really important, and I was really inspired by what you told me before, that actually this is I know it's not official, but the sense is that this is a how, not a what. That is, the company is committed to individual patient level data release, but you're trying to deal with some of the technical issues about what that means, given your consent forms and a variety of other things. I mean, I'm just trying to highlight this. I thought that was remarkable and in a really important statement. And I just wanted to, there's differences between saying culturally we're not ready versus culturally we're ready, but we're now going to have to figure out how. And legitimately, there are, believe me, technical issues here about how this gets done well and fairly and attentive to a lot of different issues.
Yeah, no, I just want to echo that. And I think even with the clinical study report, and that's what we're trying to work through, as well as individual patient data, I think it's really important to hear the patient's perspective on what they want to see in terms of their own data. But we also need to hear the patient's perspective on how we're going to protect their privacy. And the way we have individual patient data now, as well as our study reports, I'm not sure can really accomplish those things. And so we want to work towards that and I think the best way to do it is to collaborate together and really to hear all of the different needs of the different groups, researchers, patients, investigators, et cetera.
So I want to comment on a couple of things that have been said, because I mean, my understanding is the next day and a half we're going to cover a lot of these technical issues, which are technical, ethical, cultural, statistical, very complicated, and not worked out at this point, in my view. On the study reports, there are really two study reports that need to be made public. One is what the company sends to the FDA. The other is the internal FDA analysis. And I think a lot of people don't understand that right now, if the drug doesn't get on the market, these reports are not available in any form. Our brilliant U.S. Congress has passed laws that really restrict these data from being available unless the company makes them available. What? Yeah.
It's a key point, because people think that it's illegal to make them available. It's illegal for the FDA to make them available.
Right.
It's not illegal for the company to make them available.
Right. So what's in the study report, as Jesse, I think, very well described, is a lot of detail, and it's critical for people to know that what's in the journal is the end result of sometimes hundreds of decisions about what's included and excluded in terms of patient population, analysis techniques, et cetera. Those are usually covered from the company's perspective in the study report with a lot of editorial and bias. And there's nothing wrong with that. That's the company's job, is to make decisions and defend them. The FDA report, and the FDA, as opposed to the European regulators, actually get the raw data. And the penalties would be so extreme for holding back raw data that it's not often done. The FDA report is often quite different than the company's report because it takes a totally different view, not biased by the company's perspective. It may be biased by someone at the FDA's perspective. So I think those are critical things. But I think what several people have mentioned also, right now, it is the case that if you made the raw data available from these reports, they are identifiable. There are enough characteristics of people in those reports that you could identify individuals. Whether that's good or bad is another discussion, and I hope it'll be discussed during the course of the day and a half.
So these are very good segues to the following sessions. Maybe we'll continue questions from the floor. Yes, sir.
Hi, it's Jose Galvez. I just want to say first, I'm a real advocate of sharing data, both clinical and preclinical data. But what I've seen as a real impediment to the data sharing is working with the academic colleagues is that they're really disincentivized to share data. I mean, the reality is for career advancement, it's authorship, not creation of data, which counts. And so I think one of the big problems we're going to have to tackle is how do we incentivize academics to share data and allow this to count for their academic advancement?
We'll start with our journal editors.
Yes, that was why my proposal was that if you've generated data, you are automatically an author. The only thing that would prevent you from being an author would be if you disagree with the analysis, in which case you're a collaborator. You are always attached to your data, and so is your funder, and so are the trial participants, and so is everybody involved. And the journals need to learn ways to give credit to that. Just because Medline doesn't want to have huge author lists doesn't mean we don't have huge author lists. We handle that quite well, and we're developing actually markup so that we can send your actual author contributions directly to your resume. we are marking up exactly in excruciating detail that this person was the analyst on this person is the designated representative of this consortium, these people contributed equally, these bosses jointly directed the project. There should never be a discussion again about authorship order. I think we need to have really particulate, granular identification of what people actually did in knowledge production. And I think that is where the incentive comes from.
I just say that I think this is a problem in the House of Academics that we have to solve. It's not a journal editor issue. And I also think that it's unwise to think that only the people who generated the data set should be the ones who are going to be writing on it. The people who are writing on it may be at odds with some of the things that were said. There may be the way in which the public dialogue progresses, but the people who generated the data need to get credit for as the progenitors of the substrate, of the reagents that people are using. And that's a House of Academics issue. And maybe there are ways in the journal, even if they haven't written that article, that there's credit and acknowledgement in the article. This was produced by blank. Because really, you can't have done that work unless the people had put in that elbow grease, and they deserve great credit for it. And but the current thing is that the people feel ownership. They feel like they can take their time. They feel that they can do it as ever they want, and that leads to slower. Some groups are extraordinarily productive, like Rob's, who really were producing quickly and rapidly from the trials, and others publish one thing out of the trial, and it takes years and years for anything else to come out. And we've got to change this game in academics. We have got a big problem.
Yeah, clearly there needs to be a mechanism that's carefully thought through that allows credit to accrue back to the people who do the actual very, very hard work of clinical trials. And I think it's already an underappreciated business. If you do a trial and do it well, it takes a huge amount of effort, and you get a couple of publications out of it, maybe. Take the example, though, which is somewhat similar to what we're talking about, of people who do meta-analyses and systematic reviews. They churn them out, and they're all based on work that has been done by other people. So I think we already have a system in which the people who do the hard work of clinical trials are not recognized to the extent they should be. But Dr. Krumholtz is right. I think you can really lay this at the door of the academic medical establishment, because journal publications are the currency of academic promotion. And until that problem is solved, and there are ways of recognizing people not just for the number of publications they have or the impact factor of the journals in which they publish, but for the work that their work has led to, for the importance of studies and work that is done based on things that they laid the foundation for.
Labors in the fields of actually doing clinical trials. You guys have made my day because I used to go to meetings with Deborah 5 or 6 years ago where I'd be the only trialist in the room. There would be a bunch of meta-analysts who I just felt like all they wanted was for me to work for 10 years and then hand over my data set. So without doing any work, they could sit at their computer and write a dozen papers, often misinterpreting the context of the trial and the details of what the data definitions actually were. So I was also particularly excited to hear Miles's proposal that there needs to be, the progenitors here are the parents. They need to still watch over their children to some extent. Now, they shouldn't control their children in this case, but to ignore the real fundamental knowledge of why the trial was done, what the patient population was, and how the data were collected and stored is a huge mistake that I see over and over again. Deborah now, it's turnabout is fair play. Now that she's in charge of clinicaltrials.gov, we frequently talk about really stupid submissions to journals that we're reviewing that where people didn't understand how the data got into clinicaltrials.gov, and they make really bad decisions about their analyses because of that.
This was very good. Dr. Rosenblatt?
Yes, I wanted to sort of pick up on this theme of the dissemination of the information that you get by revisiting data sets, and I think this conversation has been very good at kind of highlighting some of the issues for an academic look at this. But I think one of the realities as we open up data much more widely is that, and I want to take advantage of having journal editors here, is that not everybody who's going to be looking at the data cares one way or the other as to whether it'll lead to a peer-reviewed publication. people will be going into these data sets for many different reasons, and they may not choose to go to your journals and subject it to rigorous peer review and dissemination through carrying the kind of imprimatur of some of your journals. And so we haven't at all talked about, once you get this data, how do you make it available in a responsible way, and what are the standards? I think Dr. Axton talked about that a little bit, about having a hypothesis and peer review, but how do we do that? Once we open the data set, how do we make sure that it's reported in ways that are responsible, even if they're not necessarily going through your particular organizations?
It's a very good question. Could we have let's have a quick response to that, and then we'll take one more question from the floor. So this issue of how do we ensure responsible utility of the data that's made more widely available, perhaps not just it being published in the article in the newspaper.
So I was criticizing the otherwise rather good NIH data access committees because they're slow. The assumption is that the government paid for a truckload of sand, it goes into a sand repository, and then you're not allowed to use it unless you go through all kinds of hoops. I was proposing the opposite of that, which is that the data holders have an obligation to share the data, but that they are always the generator of that data, and the assumption should be that anybody qualified with a good idea gets a chance to look at that data. If they're rebuffed, then we should have an ombudsman committee, which is partly the data generators, partly independent people, and partly research participants, to help redirect the sort of research so that it becomes acceptable use. In other words, it protects the research subjects, it has a useful research question, and that could then be accepted. So that was my model.
So I have a related question, though. That doesn't protect against someone writing, let's just say someone writes a flawed or stupid analysis that gets into some journal. And Rob is right, I have been living this a little bit with clinicaltrials.gov data. Papers can get rejected at top journals, but they show up in the news, and I know it's flawed, but I don't have time to rebut every silly analysis, just like a company wouldn't necessarily have time to be a police about the analysis of their data. Is it sufficient to just let open science take care of itself? In other words, criticism will occur, and it can occur because the data are open, or do you need some other mechanism?
Right, and the concern would be that these go on to live in the internet and never go away.
Right.
Right, so you're talking about people who maybe don't do a good job analyzing the data, and you're discussing a related problem, which is those get rejected from good journals some of the times, not always, but then they appear elsewhere in their original form, and that's something we regret to see too when we reject papers, that we make comments and then they're not taken on board. That's a problem in part, I think, that journals are well placed to solve by trying to link all commentary about a paper back to the original study, and I'm confident there are mechanisms to do that. But beyond that, also publishing peer review and commentary alongside the paper in a way that can always be linked back to the clinical trial. I do, certainly it's possible to imagine lots of scenarios in which bad analyses of data have a lot of currency and popularity among the population. But I think taking the long view, that is likely to be transient, and that we should have confidence in the fact that eventually science is self-correcting, maybe not within 5 years or 10 years. I don't think we should be afraid of having multiple competing analyses out there because, as Peter Gotcha said, we don't want to live in a society where there's only one version of the truth. And I think the anxiety that we feel about many different people looking at the data and coming up with different interpretations is somewhat misplaced. I don't think we should be afraid of that future. Now, journals will certainly play an important role in sorting through competing analyses.
Right. Well, if I could follow up, I think that the peer-reviewed journals, which are at the kind of top of the food chain, nevertheless are a smaller and smaller slice of the dissemination of information.
Yeah, I would like to make a very strong comment about this.
Yeah.
I'm very in favor of data liberation, but if you're talking about self-correcting science with inanimate objects in a lab or even preclinical science, there are not many consequences. But Much of this is going to happen outside of journals, much like paparazzi, and there will be consequences of people being killed by poor use of data. Because if it hits the news, a lot of our people will stop taking their medications or whatever it is based on what's in the news. And so you can kill people with bad science very quickly. 5 years is too long. It's a problem that we're going to have to grapple with.
So I suggest that we return to some of these themes. Since we're on the internet and the timing is very important, I'm afraid we are going to have to close this session. Thank all the panelists, the speakers, and the audience for a very useful opening session. Thank you.